Today I'd like to talk to you about periodontal disease, a chronic inflammatory disease that affects approximately 46% of U.S. adults. In our mouth, foreign microbes form a layer of bacterial cells, or plaque, and our immune system's response to this is an inflammatory response. When this gets prolonged, it causes a state of imbalance, and certain enzymes, called MMPs, are increased. They act like biological scissors that degrade the tissue surrounding our teeth. If left untreated, this leads to periodontal disease, which can result in bone loss and even tooth loss. Now, for my research, I want to learn more about this inflammatory response by studying these enzymes, MMPs. To do this, I have used a multi-cell system with an added agonist, Hag-B, a protein from a periodontal-associated bacteria. Now, this multi-cell system consists of T cells and dendritic cells, two cell types known for their immune cell functions, and then keratinocytes, which are cells from the lining of our mouth. Now, this system is unique because rather than the standard method of growing one cell type by itself, we're growing three cell types together, and they're allowed to communicate with one another because their signals can go through a porous membrane here and here. And so that way we can study the effect the cells are having on each other. So for my experiment, I grew each cell type by itself, in pairs, and then all three together. And then I added my Hag-B, and after 64 hours, I determined the concentration of 9 different MMPs produced by the cells. Here's one of those MMPs that had significant differences between the groups. So what we see here is that dendritic cells are the main responder of this, the main producer of this MMP, while the other two cell types are not. Now, when paired with either of the other two cell types, there's still a very high response coming from the dendritic cells, whereas when paired with all three cell types, we see a different response. So what this tells us is that the cells are responding differently when they're grown by themselves compared to when they're all grown together. So as we continue this research, we want to use a multi-cell system because it's going to better represent the host environment where many different cell types are present. So my lab hopes to use this multi-cell system to do 3 different things. We're hopefully going to use it to identify other MMP inhibiting products, and then learn more about the inflammatory system of the mouth. And thirdly, in time, we hope that it will help us to identify potential treatments to inflammatory diseases like periodontal disease. Thank you.